tried to isolate components of them that were actually doing the cell kill. Now, starting off, everybody knows about cancer, but not a lot of us know what it really means. It's some sort of a mutation in a cell that causes it to grow and divide uncontrollably, usually at the cost of our normal healthy cells. Now, it is responsible for the second most deaths in the U.S. behind heart disease, and a quarter of the deaths in the country are caused by cancer. About 1.7 million people are expected to be diagnosed in this next year with cancer, and 600,000 of those are expected to die. Breast cancer is the leading cancer among women, not including minor skin cancers. Now, cancer usually moves through an enzymatic pathway called the COX-2 pathway. There are two different types of COX genes. The COX-1, it maintains your normal levels of these proteins called prostaglandins. Those will moderate your cell's normal life cycle and how long they stick around. But COX-2 is an inducible form of this. It comes up usually in cancer or when you have inflammation, swelling of a wound. And usually that's a good thing, but if you have too much inflammation, it can turn into a bad thing. Or in the case of cancer, it can overactivate, and it will be constantly producing these prostaglandins, namely prostaglandin E2, which will cause the cells to continue to divide, create new blood vessels around them to supply those cancer cells with nutrients, and to activate these beta-catenin and Wnt pathways, which will keep the cells replicating and cause them not to go through their normal cell death phase. So you can see here the action of the prostaglandins coming from the COX genes and activating a whole cascade of proteins going through beta-catenin and into and activating or deactivating or activating cells genes which cause cell survival, angiogenesis, cell proliferation, like division of cells, and anti-apoptosis genes, which is that normal cell death. So there are a type of drugs called NSAIDs, non-steroidal anti-inflammatory drugs, like lots of your normal painkillers, that will reduce inflammation by inhibiting this COX-2 pathway. And there have been previous studies that showed that normal drugs that we take for pain can also have an effect on reducing cancer risks and treating cancer. So our lab typically tries to look at plants that have been used in treating inflammation, because if inflammation and cancer both work through that COX-2 pathway, maybe the things that are stopping the inflammation will also inhibit cancer if they're working through that same pathway. So looking at the plants or plant and fungus that I was dealing with, we start with the Inonotus obliquus, normally called the chaga mushroom. It's present in birch forests throughout Russia, Asia, and the northern parts of the US. Usually it only comes up after the birch trees have died. And throughout Russia and Eastern Europe, it's used as an immune boosting and cancer preventative tea, which is odd because the full names in Russia and Poland for this mushroom literally mean tree cancer. Another use of this is to be mashed up into a salve and applied to wounds in a compress to reduce inflammation. Now, Equisetum parvus, or the common horsetail, it is the oldest extant genus of plant species. It's been found fossilized all the way back to the Mesozoic era, 150 million years ago. It ranges from the Arctic to the temperate regions of the northern hemisphere. There are places around here that we can find horsetail. And in Japan and Korea, they eat it as a vegetable, sort of like asparagus fronds. All the way back to ancient Rome, they have been using this to treat wounds and reduce like skin rashes and diseases. It's also been known to treat disorders of the locomotor system, kidneys and urinary tract, and rheumatism and gout through the use of a tea. So, what I did was basically going through and there, it was a bit out of place, but our process is basically a where's Waldo of looking, and you see you have this big pile of crude things that you know is doing something, but you don't know exactly what in it is doing that thing. So you have to try and narrow it down. If we're looking for Waldo, we have to think, okay, first I'm going to look for somebody in red. Then you can filter all of the other colors out. Then you can look for someone wearing a hat, then someone wearing glasses. And eventually you're going to narrow it down enough where you can find your Waldo. So first thing we did was obtain our crude extract. I took 25 grams of the powdered horsetail or chaga that we purchased from a culinary company online to ensure that all of our samples were heterogeneous, even though we could have gone and collected some of these in the field. And I refluxed those in 500 mils of ethanol or acetone in the case of the chaga for an hour and a half, and the crude extract was collected. I settled on those because prior to this part, I did a panel of several extracts and tested them to see which ones were doing the best cancer killing, which was extracting the cytotoxic components the best. Now, before I could put this into the next step, I had to distill the chaga from acetone into ethanol. Now, the next step was to run a Cefidex LH20 chromatography. This is a size separation. So imagine a bunch of tiny balls of Swiss cheese, all packed into a column. Now, when you drop your chemicals into the column, the big ones are going to run around and through and come out of the column first. Now, the smaller ones are going to get caught in those holes inside the balls of cheese. And they're going to take a little longer to come out to the bottom, right? So what this is doing is giving me larger components on the bottom coming out of the column first, and smaller ones coming out last. So I collected 200 fractions of volume of 5 milliliters each, and then I tested each of those samples for absorbance at 280 nanometers. This is the wavelength where lots of your smaller proteins and your molecules with conjugated bonds that will not always refract visible light, but they can usually refract in this range. So this was just seeing if I was actually getting something in these samples before I moved on to the next part. Now, from here, I did a cell glow assay of these fractions. I took all of them, and each one I put into a well on a plate. Those plates were treated, or I had previously put cancer cells into those plates. I treated the cancer cells within each of the wells with a microliter of fraction per 100 microliters of media. And the next day, after they had incubated for a full 24 hours, I tested them to see which things were causing killing within the assay. So, moving on to this, you can see here my absorbance spectra of these fractions. You can see down in the beginning, where later on this will be significant, I was seeing most of my absorbance. There were small peaks around here and around here with the horsetail, but chaga was mostly, pardon me, right in this beginning area. Now, looking at this cytotoxicity assay, now this one was more of a qualitative assay. I was just looking to see where the cytotoxic components were being located. So as long as I was running this, they were going to inevitably be stretched over more than one sample. So what I was trying to do is see which areas these samples were coming out of. So looking at those, I could see, well, right in here, there's several things doing killing, so they're probably all the same thing because they're all coming out at the same time. So what I did was I took all of those fractions and pooled them together and called them a band. So I had 5 bands coming off of horsetail, and if you look here, the peaks around A and B were also places where I was seeing absorbance at the 280 nanometers. So that's just more affirmation that I'm getting what I'm thinking. Now, chaga was a little bit easier. I only had 2 components come out of chaga from this, and I pulled them together and had 2 chaga bands that I was going to test further. So after I got these bands, I put them into an HPLC, which is another column, but instead of it being those little balls of Swiss cheese, it's, think about magnets. You have, all of these molecules are going to be some sort of polar that I'm getting, probably. They're either going to be really, really polar or nothing at all. And in the column, you have like little balls of like long threads pulled up into something about the size of a pinpoint. And all of these have molecules sticking on the end that are some degree of polar. And we're trying to use a solvent to move them through this column. So with this one, I'm separating by their polarity instead of size, adding another sort of filter to what I'm doing. So these also would come off with a spectrophotometry reading. And you can see here, I was getting a single peak, so probably 1 molecule coming out of my HPLC. And I saw this peak here. There were 2 peaks in band C. Band B did not show any cytotoxicity in horsetail when tested preliminarily before this, so I did not include it here. Band D showed another peak here, although it's a very small peak compared to A, with an absorbance of about 500 nanometers, or 500 milliamps, or milliabsorbance units, sorry. And band E had a similar peak, but again, the absorbance was not as much as was in A. So from here, I ran another cytotoxicity assay, this time in octuplet. There were 8 of each of these components that I was testing. And I analyzed them with a one-way non-repeated measures ANOVA. Now, looking at all of my chaga bands, we only saw killing from chaga band B, and the only peak I saw in it, which I guess my things have gotten out of order. And the horsetail assay showed cell killing in all except for band B, but most of all in band A, where I was seeing the biggest peak. Now, we sent these off for a mass spectral analysis, which is just telling me how big my molecule is, like actual quantifiable number size. And going through, you can see the labeled peak here at 412. The way the MALDI works is it shoots a proton at the molecule that you're testing, and usually that proton will stick to it and add 1 mass unit to it. So it's about 411, not 412. And you see nothing in band C, or the band C peak 1, meaning either I didn't have enough like concentrated extract when I sent it off, or it was some sort of an anomaly from my other tests. Now, looking at band C peak 2, there's another peak on the mass spec at that same weight. And again, in D peak 1, and again in band E peak 1. So we're thinking that either these are all isomers of the same thing, slightly different arrangements of all of the same or all of the same components, or I am getting one single thing that is coming up in my entire sample. So we had to go back and run more HPLCs, and here is a mixture of band A and peak 1 and band C peak 2. And if they were isomers, I would expect to see 2 separate peaks coming off of here. But I did not. I saw 1 peak here, and it is way higher of an absorbance than previously seen in our HPLC of the crude bands. Now, looking again at mixing band D with peak, or band D with band A, same peak, same spot at about 32 minutes. It's like 32.3. Same thing, band A and band E mixed together. Now, when I look at band D and band C, peak 2, you can see here that the absorbance is a lot smaller going along the axis, but I'm getting a small peak at the same point. And again here, and again here. So all of my mixtures were coming off with that same band, although it was much higher in everything that had band A in it. So the known molecules that this might be all involve cobalt. And thanks to Dr. Allen, I found that horsetail is a cobalt fixating plant. They have bacteria that take nitrogen from the air and move it into a molecule, and move it into a form that the plants are able to use. But these bacteria need cobalt to function, and the plant supplies the bacteria with cobalt. So it is likely we will need further testing to confirm if cobalt's there or not, but it is somewhat likely that it's one of these different molecules here. Now, going back to the chaga, there was no results from the only cytotoxic peak from the chaga, which probably means, again, that my extracts were not concentrated enough, so it will require further testing again. Now, I also did a toxicology study on these. I acquired 12-hour stage zebrafish embryos. We use zebrafish for these sorts of things because they develop clearly and quickly, so you can see all of their stages of development. And you can watch, I have a small video that lets you see where you can watch the heart actually beating and the blood moving through all the chambers of these. We incubated them at room temperature with a standard concentration of sea salts. I used 100 mils of crude ethanol horsetail extract and concentrated it down to 20 milliliters because ethanol has this funny thing that it does with embryos where it turns them into cyclops. So that would not be something that I wanted to happen. I took these and I diluted them into 1 to 100, 1 to 1,000, and 1 to 10,000 solutions of extract to the zebrafish water. And I had groups of 27 embryos per solution and 30 embryos in my zebrafish, or in my control. And for the next 11 days, I recorded their death numbers and mutations. I also did a set of mouse studies with Sarah Kelly. If you look in the hallway, she has a poster doing a similar project which you should stop by. We injected them with cancer, and then for the next 2-3 weeks, we injected them with our treatment to see if it would do anything to it. Unfortunately, at the end of the experiment, we were not able to record metastases as we would have liked because the tumors were not detectable on their lungs and livers. We had, okay, so in the zebrafish toxicology, we had only 4 deaths occurring through the entire process, and oddly enough, they all occurred on the same night. But there were no mutations seen throughout the entire process, even in the fish that died, which is a good indication that whatever is killing the cancer is killing it selectively. Our mouse trials were somewhat unsuccessful because we weren't able to measure their metastases. It took away from a dimension of our project, and we saw no real difference within their tumor size, which was unfortunate, but that's probably something we're going to try and redo and see if we can modify to make it any better. We concluded that we have isolated some cytotoxic components from the horsetail, and the horsetail compound is ubiquitous within the extract. It's occurring through this entire thing because it's so prevalent. The horsetail treatment is selective for the cancer based on our toxicology report and may contain cobalt, but we're going to need more identification process, or more identification and more in vivo studies added. I'd like to acknowledge the Welch Foundation, Dr. Moore, Dr. Allen, Dr. Hahn, and all of my group that helped me with my toxicology report. And thank you.
Any questions?
Yes.
Jake, you said that the light you had on there was not visible. What kind of light was it?
It's ultraviolet.
It's ultraviolet.
Yes.
Was it contained within your device, or?
Yes, we have our spectrophotometers shine a certain light through the sample and measure refraction off of it. Yes.
I know you kind of stumbled on the cobalt thing kind of recently, but have you had a chance to see if there are any other anti-carcinogenic compounds or drug treatments that involve cobalt?
No, well, I have looked in the literature, there are none at all. Most of the compounds from like plants and stuff like this are a lot smaller, and they're usually phenolic compounds. Cobalt, those three compounds I have, basically the only thing we know about them is they exist. I have not found anything in any sort of literature about them being used for anything at all. There are some studies using B12 in cancer, but most of it is using radioactive labeled cobalt to measure where that molecule is going and labeling it.
Do you know of any metals that are used in cancer treatments?
Not that I know of.
So why did you hire a lot of cobalt for the molecular weight or
Molecular weight, yes. It was
that's the way.
because of all the compounds that I found being so very closely related in molecular weight. The only thing that came up that was close to that was the cobalt, those three cobalt-containing molecules. Yes.
Okay, so you did a comparison with the NSAIDs to your horsetail and frog extracts. Was the data that you found significant in any way to that comparison?
To the comparison, there have been some things that have used like Aleve and stuff and other insects for some small cancer treatments. Usually what the people find with that is that it's kind of ineffective, like we have found in our cell and molecular biology class. Most of your common NSAIDs aren't quite strong enough to affect the cancer directly, especially when you're taking the normal recommended doses.
I just didn't know if like the dilution that you used for your horsetail and frog was like significant in the same aspect of what you would take on a going basis.
Well, since we don't know what the molecule is, we don't know what concentration we have it at. It's just basically the concentration that we can get from that specific amount of plant with the best solvent that I've been able to find.
Well, let's thank our speakers.